For our big experiment in the history of biology, we decided to look into the sickle cell disease. We will be talking about the hypothesis that was tested, the experimental group, the results of the testing, and what are the long-term implications. We will also have two multiple choice questions, and I expect for you to answer those questions. So thank you for watching, and I hope you learned something from this video. So the first modern report of sickle cell disease may have been in 1846, where the autopsy of an executed runaway slave was discussed. The key finding was the absence of spleen. Reportedly, African slaves in the United States exhibited resistance to malaria but were prone to leg ulcers. The abnormal characteristics of the red blood cells, which later lent their name to the condition, was first described by Ernest E. Ahrens, who was born in 1877 and passed away in 1959. Another professor of medicine, James B. Herrick, he saw peculiar, elongated, sickle-shaped cells in the blood of a man named Walter Clement Noel, a 20-year-old first-year dental student in Granada. So, large-scale natural history studies and further invention studies were introduced in 1970s and 1980s, leading to widespread use of prophylaxis against pneumococcal infections, amongst other interventions. The 1990s had the development of hydroxycarbamide, and reports of cure through bone marrow transplantation appeared in 2007. In the US, there are very large variety of stigmas around sickle cell disease that discourage people with sickle cell disease from receiving necessary care. These stigmas largely affect African Americans, since the majority of cases of sickle cell disease in the US are in the African American population. People with SCD experience the impact of stigmas of the disease on multiple aspects of their life, including social and psychological. Studies have shown that those with SCD frequently feel as though they must keep their diagnosis a secret to avoid discrimination in the workplace and also among peers in relationships. In the 1960s, the US government supported initiatives for the workplace screening for genetic diseases in an attempt to be protective towards people with SCD. By having this screening, it was intended that employees would not be placed in environments that would potentially be harmful and trigger SCD. However, in practice, this screening was discriminatory and allowed for employees to place restrictions on employees with SCD and carriers as well. So please continue watching because we have more information for you. Thank you.
Hello, my name is Wendy Salazar. So the purpose of the experiment was to report the difference in electrophoretic mobility between hemoglobin from healthy individuals and those those with sickle cell traits have a mixture of the two types. The difference in electrophoretic mobility is probably due to the different number of ionizable amino acid residues in the protein portion of hemoglobin, and that this change in molecular structure is responsible for the sickling process. It also reports that genetic basis for the disease, those with sickle cell anemia are homozygous for the disease gene, while heterozygous individuals exhibit the usually asymptomatic condition of the sickle cell trait. The experimental groups that I found was the Linnaeus Pauling was a prominent physical chemist at the California Institute of Technology in the mid-1930s. Pauling turned his attention to the physical and chemical nature of hemoglobin, and in 1946, he sent graduate student Harvey Itano the task of finding differences in hemoglobin that might explain sickle cell disease. So after failing to find any differences in size, weight, or acid-base titration, Itano found that the oxygen could inhibit the sickling process while various reducing agents could speed it up. This was the basis of Pauling and Itano's first publication of the disease. Itano also found that the globin portion of the sickle cell hemoglobin had a barely detectable difference in electrical charge. So the experimental group is the hemoglobin, and I don't think they have a control group.
Good afternoon, class. My name is Yerbeli Sosa, and today I'll be talking about the sickle cell anemia disease. So with this disease, they decided to do an experiment to see exactly what was going on with the cells. And today I'll be talking about the results of that testing. So whenever they did an experiment, they came up with the results that they found out the blood samples from sickle cells anemia patient and normal adults reacted different in two ways. So whenever they did this experiment, they didn't only find that it reacted in one way, but it also reacted in two different ways, which is kind of crazy, just because whenever you do an experiment, sometimes you only get one result, not two. But the first thing that they saw was that whenever they graphed the curves of the mobility versus pH, the sickle cell hemoglobin curve followed the same path as a normal hemoglobin but had a higher acyclic point. The second thing that they found was that sickle cell and normal adult hemoglobin behave differently when their carbon monoxide derivatives are subject to electrophoresis at a neutral pH. Something else that they found out whenever they were doing this experiment was the sickle cell anemia hemoglobin has 2 to 4 more positive charges than hemoglobin. The test also showed that the hemes are identical. Something else that I found interesting whenever I was doing my research on this experiment that I didn't think that it actually could happen was that it could be inherited. So let's say that myself had this disease, and I decide or all of a sudden I have a son or daughter, it could be inherited. So if I have this disease, my son or daughter could possibly also have this disease. So the second thing that I'll be talking about today is what were the long-term implications of their research. So when I was doing research on this experiment, it was kind of hard to find the implications of this experiment, but one of the implications that I was able to find out was that there was a significant burden significant economic burden of disease. But it kind of makes sense. If there's a disease going on this bad, then it can hit our economic really bad. And well, that's it. Thank you.